Thank you very much. Thank you for the invitation. So I was really curious of coming here, and yesterday already there were nice talks and nice talks about this high squids and also discussions. So I have learned a lot. So this is now a view of one neuroscientist. What are signals and noise in MEG recordings? And I put there now quite old papers, old data, because they somehow remind me very much of the state what you now have with single channel high TC squids. So why we do MEG? We do use MEG because of timing, and in fact there's in this neuroscience community there's now discussion that neurodynamics is a focus of post-connectome science, when we already have somehow quite much data about the connectivity of the brain and anatomy, anatomical things. So we should also understand the multiple timescales in human brain function. So then we already learned a lot about MEG, so why not to do EG? And we heard also some answers for that, that this is my answer. Then I go back to one of our experiments that we made in early 80s. We stimulated lower limb nerves, peroneal nerves, and then we recorded the magnetic field patterns. And then what we saw something what other people had not seen earlier. They had seen only these responses over the primary somatosensory cortex, but we saw something else. We saw also signals from the second somatosensory cortex. And the reason for that is that if you think about the sources, if stimulate the lower limb nerve, so you get activation of the foot region at the top of the head. Then you get also on both sides of the brain in the second somatosensory cortex. And if you just look how the magnetic field patterns look like, they are very local over each of these source regions, because they are more tightly spread than the EG potential distributions. And if you compare then that with this EG pattern, so this is simulated pattern, but it's very correct. So happens like that, that you get the largest positive potential here in this part. And if you would have this EG recordings, your first guess would be that there's a source underneath, and it's not. So here we get much better first guesses, and then we can go further. In fact, one of the, I think, benefits of MEG over the years has been that it has largely also facilitated and stimulated the analysis of EG signals by pinpointing these certain source areas. So signals and noise, that was the topic of my talk. So here's one noise picture from 2000, already from our previous measurement site. I did not find now a good one from our current one. But what we have here is first the instrument noise without any subject. So it's something under 10 femtotesla per square root hertz all the time. But there's a somehow environmental noise which somehow starts here at about 20 hertz. And then when there's a subject inside, we see that there's a just resting subject, there's a peak at about 10 hertz, and there's lots of low frequency stuff going on. And then when the subject is cleansing, in this case, I think just hand or the, in fact, I'm not quite sure that whether it's face muscles, there's a like artifact from muscles. And now, while coming here, then I read some of your papers. I found this by Feli et al. This paper, 2012, showing on high TC squid noise, and in fact, it's quite nicely such a. In fact, it's really nice. I was surprised how good it was, because that if we would use now that kind of sensor for recording human brain rhythms, it certainly would be that these brain rhythms, this noise would dominate. If you have 30 hertz plus 10 hertz noise, so the total noise, as this noise is this, sum as squared would be something like less than 32 hertz. 32 femtoteslas. And here's then something what of course, we should be very interested as well, because these spectra end at 1 Hz, but we're also interested at lower frequencies. This is a spectrum I got yesterday from Matti. This is from MEG electroneuroMAG case, and now in this case we have linear scale here, so it's a little bit more complicated to follow, but we see that at about 1 Hz we start to have this 1 OF noise, but still at 0.1 Hz the noise level is something like 30 femtotesla per square root Hertz. Okay, so it tells us. So now going back to old times, so because we saw already from Justin, we saw several evoked responses and that kind of recordings. So this was our situation this was from 1982. So these are all data now over 30 years old. In fact, it was me whose limbs were stimulated with electric pulses, and then we didn't have any device to check where the sources are, so we used our knowledge about anatomy and physiology of the human brain, and we knew that, okay, foot regions are in the middle of the head, so that if the sources are in the fissures, so they should be something pointing like this, and then we went along this line, one location at a time, and to somehow get a little bit more data, we always stimulated alternately left and right side. So you see that there's responses that have opposite polarities, because the dipoles are opposite there, and then we see also, because these are axial, first order axial gradiometers, so we see also polarity reversal between anterior and posterior sides. And even we see that it's a slightly different locations for left and right hemisphere. And then we heard today that the best thing to know what is signal and noise is to look at the noise, what Alex was saying, but I think very important is you always make replications every time. Like, look how your signals look like from time to time. That's the most reliable thing to check this. And even while averaging, just make them to 2 boxes all the time, so they see that it's some over the. So it's really easy and quick. Okay. Then yesterday we saw also like plans to make a 7-channel. High TC, this was our 7-channel device from 1986, and then here, for example, we have put it to 2 locations. So again, it was like first order gradiometer, so axial gradiometer. So there's a polarity reversal between these 2 locations for this early 20 milliseconds response. This nomenclature is typically, so we have taken this that it says that this N means it refers to the electric potential and its polarity. So its polarity is it's negative on the scalp, and this refers to magnetic. So it's magnetic counterpart for that. So it took quite long time to take this whole head coverage. This is already a multi-channel device in '84. And then 7-channel, 24-channel, it already had a planar gradiometers. And then first in the world, whole head coverage, 92 or 93, 92 it already came, and then later this electroneuroMAG device by a spin-off our research team. And so we need this whole head coverage for effective artifact removal, as I will show you later on. Also, to really get reliable signals, because we can use these single channel devices just to detect some evoked responses. But if you would like to study some cognitive functions or brain rhythms to know where they are located, you need multiple. That's a multi-sensor device. And then we also had, as we saw yesterday, so all kind of wooden gantries over the year. And then another thing has been all the time to how to then know where your single channel device is, with all kind of very strange grids. And then, for example, this is like just a spot to target then the tip of the viewer to the certain location, and then have a grid. And then, of course, nowadays this type of 3D localizer that makes life extremely easy. So with these new devices, you will get the field pattern, or that maybe in 15 minutes, similar field pattern that earlier would have taken weeks to collect, because then you also had to repeat it several times to know that the subject's state was still the same as it was a week ago or something like that. Here the median nerve has been stimulated at the wrist, and then we see very early responses at 20 milliseconds in primary somatosensory cortex of the other hemisphere, and this second somatosensory cortex is activated in both hemispheres later on. So this is the how I think the state-of-the-art responses look at present. And then about auditory responses that we saw also yesterday in a couple of talks. So this was our first response for this N100M, so the counterpart of this 100 millisecond response that is negative here at the top of the head. And this here the I will come back to this interstimulus interval, but in this case it was 4 seconds, so it's important for the responses. And then there, in this case there were still very many responses, averaged over 300, and this in this picture it's important again that we start at 0.03 Hertz, and then this has been low pass filtered, and the source seemed to be the auditory cortex in contrast to what had been earlier thought. Now again, it's important that depending on your filter settings and the number of how many responses you average, you may get quite a different answer. Because in all evoked responses there are early responses and later ones, and they behave in a different manner. So if you have a wide band and you can record several, you can average several responses, so you will see even the very early cortical responses, and then otherwise you see these later long latency responses. Now I mentioned the interstimulus interval already, so it's really an important factor. Here we have an interstimulus interval from 1 to 16 seconds for both magnetic and electric responses to tones. And you see that there's a very clear increase in amplitude when the interstimulus interval is increased. And here I show the same thing for visual stimuli. So as a function of ISI from 1 to 4 seconds, the early responses in early visual cortices, they somehow saturate already at less than 2 seconds interval, but in some other areas they saturate only at very long ISIs. And you can compute here, you can model this as an exponential function, and then you get this some kind of lifetime for this for this behavior. And if we see that the tau lifetime is here less than half a second, and here it's over 10 seconds. So it varies, and it's better to take that into account also. So this is just the curves fitted there, and so this lifetime is the longer, the higher is the source in the processing hierarchy. That is one such a very common thing. So when you go from primary areas to some higher order areas, this gets longer. And the other thing, tau is the longer is the latency of the deflection in the certain response. So that early responses are not so sensitive to interstimulus interval, but the long ones are really sensitive.
Sorry.
Yeah.
Yes,
in this is its amplitude.
Why don't you look at the phase response to the signal? Phase shift versus phase response.
Yeah, okay, so that is because we stimulate, okay, we take, these are like transient responses in this case, because we present them very unfrequently. So it's something, you get a response which looks like this with the 8 seconds interval, and then with, so you get very similar but smaller one here at the short ISI. So we can look at the peak latencies here. There are some, okay, let's say in EG responses, there are very clear changes also in the latencies, peak latencies. But it's not just like, I don't think the phase in this type of case would be the best thing to look, but it's better to look at the latencies of these different parts of this response. And it's here it's also, of course, then also possible, for example, for clinical purposes. Knowing this ISI dependence, we can find the optimum interstimulus interval for certain response, for best signal to noise in a certain measurement time. So because sometimes, for example, we would not like to keep the patient too long there. Okay, so then about the frequencies, the relevant frequencies from neuroscience point of view, they range from DC, or let's say at least from quite low frequencies, up to hundreds of hertz or even over 1 kilohertz, as we heard just earlier. And here you just see that, for example, here the bandpass has been up to about 150 hertz, and we can separate clearly and replicable manner, as you see there's replications of the same measurements, several deflections here in this evoked response, and then they of course have also clinical importance. And in this case we have presented very long sounds, emotional sounds that have lasted each of them from 2 to 8 seconds, that is 6 seconds altogether. And this is a DC recording, and you see that somehow this sustained field stays there all the time. And in fact, this is a simple thing, but many people neglect it often, that it's in fact this filtering. So even if you go to 1 hertz or even higher, so it really distorts the interpretation, because then the users don't necessarily understand what it means that you have filtered the signal. So if this is simulation again about auditory responses in the auditory cortex, this is the field pattern, we have it somehow, this is the original time course given to that with no filter, just a monophasic response. But then when we high pass it very heavily, here in this case at 6 hertz, which is of course too much for any reasons, but anyhow we see, as expected, there's somehow extra transients on both sides. And then when people start to then look at the field patterns, so they see that, okay, already here at early time there's a clearly dipolar pattern in the brain, must be that this response started at this and this time. So I think there's wrong interpretations about response latencies, for example, because of these ones. So I presented several evoked responses because in the beginning they were the easiest ones to record, because you can stimulate several times and then you can average the responses. And it's much more complicated than to look at the spontaneous activity, because you can't average that. And this was also mentioned yesterday. This you mentioned both the alpha and mu rhythm. So this is just from this early whole head coverage in early 90s. Spectra in different parts, so there's always for these 2 planar gradiometers. So at the back of the head we have this alpha activity with main peak at about 10 to 13 hertz, suppressed by opening of the eyes. And then here we have this mu rhythm, which is somehow has a peaks at about 10 and very clearly typically also around 20 hertz. And this mu rhythm is dampened, for example, it's not affected too much by opening of the eyes, but moving the left hand or moving the right hand abolishes this. So here I would like to emphasize 2 things. Maybe first that the sources, if you follow single spindles, even with very good signal to noise spindles, so typically these one single spindles only live less than a second, something like 600 milliseconds. And then there seems to be like another source. So there are lots of sources for this in the parieto-occipital part, and then a little bit also in the visual cortex. And there's intracranial recordings support us as well. And then in the mu rhythm, this is mu rhythm typically has this type of archaic shape, which already tells that it can't be just a single frequency. So it has 2 main frequencies, one around this 10 hertz and the other 20 hertz. 10 hertz seems to be generated a bit more posterior than the 20 hertz, so we think that mainly 10 hertz reflects somatosensory processing and the 20 hertz mainly motor processing. Okay, so then signal responses, evoked responses, they are very small and prone to artifacts. I presented this earlier, these responses that were averaged, but we would like to, in many cases, look also at the signal responses. Here is just an example how the responses look like to phases that are presented for different durations, starting from 17 to 200 milliseconds, and their phase is coming, then a mask phase is coming, and these are signal responses time locked to the onset of the stimulus. And you see that here you can't really see this response coming, but here we see something like enhancement of the activity and then followed by a different polarity response. So you need a certain duration of the stimuli to get the reliable responses, and still they are very variable. And then somehow the problem is that we have such very strong enemies. We have multiple enemies which are all kind of artifacts.
Sorry, what was on the y-axis? What the number of channels? So 1 to 18?
No, one. This.
Right.
Yes. Here, this is just a single trace. Like
Okay.
it's presented once. Yes. Sorry.
For each channel?
It's
85 or
yeah.
90 channels?
yes. So, yeah.
No,
No.
trials.
The trials. Yeah. Sorry. Trials. Yeah. Trials. Yeah. Yeah. Sorry. Yeah. This is like a single position, single channel, and this just shows the same information, but one after each other. Yeah. So the enemies are here. So we have all kind of artifacts from, for example, eyes are strong dipoles that will cause us eye blinking and eye movement artifacts. Then we can have muscular tension, the respiration-related stuff. We have also magnetocardiographic artifacts and also ballistocardiograms. So if you look, ballistocardiogram is something which is related to the somehow mechanical function of the heart. So if you, for example, have some magnetic material in the body, and that during each cycle there will be a bump, as we saw recently in our common things. So that this is the cardiac cycle. We see that this ballistocardiographic artifact comes always with the latency. It's something like 100 milliseconds after this really this cardiac contraction. And then once this was just a long time ago, we had a child in the magnetic shielded room, and then we got this type of artifact because the father had a digital watch with But anyhow, so then what we should do, that we should try to prevent artifacts, and then if we can't prevent, we should try to eliminate them. And thus, I consider this introduction of this temporal signal space separation by Samu Taulu one of the very important contributions to this field, because with this new system, artifact removal system, it's possible to record also people, patients who have some pacemakers in the body, as earlier was totally impossible. And somehow the point is just like based on just the basic principles of basic properties of magnetic fields, and the fact that we now have almost whole head coverage. So of course we are still missing a large part of this sphere that we are not covering all the time. But it now turns out that it's possible to discriminate signals that are coming outside of this area and inside of that. So then of course we are interested only about signals that come from the brain. And here's just one example. So this patient has a deep brain stimulation, and now we have 30 seconds of MEG, and it looks ugly. So it's just full of artifacts all the time. And then, as Matti says often, that before treatment and after treatment. So this is before treatment and then after treatment. So it's a beautiful thing as we see all the occipital. Okay, I didn't say that these are different channels, just unaveraged. Frontal regions, then top of the head, then back of the head, and electroocculograms. So now we can see even the alpha rhythm very clearly. That was impossible to see in the earlier data. But now, at least I was still somehow not totally convinced, because in this case, of course, we did not know what was the golden truth behind. So we just got very beautiful data, but maybe we lost something important, or maybe we brought something else. And that's why we with some that kind of experiment that we say that, okay, let's have a look at the single auditory evoked responses, because we now know quite well how they behave. And then we produced all kind of external artifacts. So this is one channel, MEG, and then from time to time, at all these times, we have presented these tone pips. And from time to time, you can see that there are really single responses, not each time. And then we caused some, we have rotating magnets and stuff like that in the close to the subject, such as toy magnets. And then we got that kind of artifacts. And then now again we have like trials, single trials here, as in earlier. So plot it like that. This is control case. So we see that there is something happening at this time when this somehow the auditory responses should appear. This is the external artifacts case. This is a nearby artifacts case. They're really ugly. And after treatment, it looks quite nice. So we can see the response here. And then if we look at the single unaveraged responses, superimposed, so they look something like this. So we were able to recover them quite nicely. And the sources go to for this each unaveraged responses sources go to the auditory cortex. Okay. So what we need in MEG, we need also, like I would say, the reasonable localization accuracy. This is not a method to really pinpoint like millimeter differences in the brain. But we need some reasonable localization accuracy and source differentiation, so that we have to know that if the sources are clearly different. Now we have one problem, is that you get what you ask for. Because if you put this type of distributed source, you simulate that in the auditory cortex or a local current dipole, so the field patterns are exactly the same. And then if you use current dipole model to explain this, so in both cases your answer is that you had a local current dipoles. Or if you use a distributed minimum norm estimates, your answer is that you have these ones. So you had to keep this in mind, that the method of analysis determines quite much what you get. Here's one example from in fact from Matti's work, but so it just also tells us that automatic search methods may bring surprises, especially for correlated sources. So these are real, I don't have this data here, but I just saw that they are like a left and right auditory cortices was activated. I understood they were real auditory data, and these are from 4 subjects. And then they applied the new method which was like invariant noise. What was the real name? In ENF, how it's called? Invariant. No, he even doesn't remember. Noise invariant. Yeah. Anyhow, so.
First, M comes from invariance.
Okay. Yeah, invariance of noise space. So. Just okay. In this case, so they were able to find sources in the left and right auditory cortex, as it should. But then there's certain algorithms that are commonly used, like a music algorithm and rap music, beamformer. So there's single activation somewhere. So, so what happens here? So is that we can have some kind of pitfalls in reconstruction. So long time ago in this tellus, there was this type of interesting animal, and then years went and he died, and then came this archaeologist and they took the bones and then they reconstructed this one. So they found the pieces, but the reconstruction was a little bit different. And then somehow the lesson is here that I think one should always keep in mind that somehow that are the results anatomically and physiologically feasible, and also that extraordinary results require extraordinary evidence. So if you find something which is totally new, others found something else, but you find it's totally different things, so you need some more information about that. So that's the okay. And then what is very interesting that this high TC squeeze could be put very close to the scalp surface. Of course, this topic has been also discussed here earlier, but this was my picture from a long time ago, some simulation, that if we have two dipoles here, then if we are able to record it just on the surface, so we see that along the surface the magnetic, the strength of the magnetic field changes like this. But if we go just 2 centimeters out, as we do with our current devices, so we get this type of nicely looking signal. So it's like if we would see this field pattern, it would be like a field produced by single dipole, but in fact it was by 2 dipoles. And we can't make the distinction unless we have extremely good signal to noise ratio for this current data, so that in fact this pattern differs a little bit from this one. But would be much better if we could record it directly from the scalp. Okay, so then we have all these problems. So could we still improve by some clever experimental procedures this our understanding where the signals are coming from and how they behave? And at least 3 things come to my mind. For example, first is frequency tagging, then using probe stimuli, and then using external reference. And first, frequency tagging is that you put some extra frequency to your stimuli. Like here, for example, you know this famous Rubin's face picture. So typically people at a certain distance, this percept changes at each, let's say, 5 seconds or something or less. You see, sometimes you see the face and sometimes you see the faces. But if we would like to look at the brain correlates of that, so then if we would ask person to always indicate, okay, now it changed, so it's far too slow a trigger for our analysis. So that's why we have put here dynamical noise. And it's, in fact, although you probably don't notice, it's of different frequencies at these two. I can still put it. Okay. So we have 12 Hertz here and 15 Hertz here. And what we then did, we just recorded with we were letting our subject to view this and then indicate us when the percept changed, and we saw in the brain clearly responses to these tags at 12 Hertz and 15 Hertz. And then we just wanted to check whether the relative strengths of these 2 frequencies change. And here now we see the main frequency at 12 Hertz when the person says that, okay, I'm seeing the vase, and the vase was had the 12 Hertz dynamical noise, and then it changed to faces, so the dominance changed. And so this was the possibility then to show what happened in the early visual cortices. So,
sorry,
yeah.
Are you talking about that you kind of manipulating the noise, or you trigger a signal?
No, we have just noise. We have dynamical noise all the time, to which we can then record these very clear signals from the brain, at this tagging frequency. Then we don't manipulate noise at all. We just ask when the subject feels that now, okay, now I have perceived the face, and then we look during that period what happened to this relative strengths of these 2 frequency peaks. So that's some, yeah, please. So in this case, we have not done different frequencies, but in auditory modality, we have used frequencies at about 20 Hertz and 40 Hertz when we were tagging information coming from one ear to the, because if in auditory system, when you get information to one ear, it goes to both hemispheres. And then if you record MEG over one hemisphere, you don't know that which part of that signal has arrived from either ear. So we have tagged them then with, in the original work, at frequencies around 40 Hertz. Okay, so then the other thing to somehow go around all this is to use probe stimuli. So if you again have some very slow phenomena, so you can send some stimuli to the brain and look, try to use them as probes of the functional state of the certain area. And like in this study, we used median nerve stimuli delivered to these wrists as probe stimuli to explore the functional state of the primary motor cortex. Because we had noted earlier that if you present the stimulus, so within half a second, 20 Hz activity that I said it mainly comes from the primary motor cortex is enhanced a lot. And then if the subject is just, that happens in this resting state, if the subject is just moving a small object with the fingers, that means activating her own primary motor cortex. So we see a very radical change. These rhythms are gone. So we use this amplitude then as an indicator of the functional state of the primary motor cortex. And in this case, for example, we've been showing that if the subject just views another person doing actions, there's a clear suppression of this rebound, as we call it, telling that the primary motor cortex was activated. And more recently, we have also used some kind of probe stimuli here, tactile stimuli with the fingers, in a subject who was viewing a movie. And we were interested at whether the haptic content, the tactile content that is in the movie, could be seen to affect the somatosensory cortex of the viewer in a dynamical fashion. And then it's impossible to somehow, at least we could not figure out any other ways to do that with MEG. So we delivered the stimuli, and then because we were not able to average this stimuli across the movie that lasted some 15 minutes, so we averaged in a way them across subjects. So we found the most common component across subjects using this multi-set canonical correlation analysis. And then we came up with responses to the single tactile stimuli, and then we correlated that information then with the rating of the haptic contact that the subject gave later on. And then we found that, yes, in the early somatosensory cortices, there are areas which indicate that the functional state of the somatosensory motor cortex fluctuates in a fine-grained manner during observation of changes in the haptic contact. And then still finally this third one, external references. We can use some external signals, for example, in this case, accelerometer put to the fingers, and then correlate, compute the coherence between the external movement and then brain activity, as what we call corticokinematic coherence, and much of that is done in collaboration with Xavier, his group, and others in our lab. And this, in fact, seems to be now the first such a very reliable measure for proprioception. So that's a new tool for that. And we have also used that so that we had an accelerometer on the throat of a person who is reading natural text to the subject. And this has been in Xavier's lab. And then we compute the coherence between the listener's brain activity and the speaker's voice. Voice is especially the fundamental frequency of that. Works very well. We see the spectra with peaks at about half a hertz, which somehow are related to the sentence length, and also at 4 to 6 hertz, which is related to the syllable rhythm. Okay, now I will show you one complex experiment at the end. Then after that I will still have one experiment, but just to show that how we can then combine many of these MEG methods. So I showed you already that when persons are just viewing another person to making some actions, their own primary motor cortex is activated, telling like that they are mirroring other person's actions. But then we can ask that how can it be that my motor cortex is activated when I see you doing something, but I'm not imitating you. In fact, some patients do imitate, so if they have some frontal lesions. But this been a long-lasting question, and then we decided to do it like that we used as a tool the Cortex-muscle coherence. So we know that if person is keeping a steady contraction, the hand for example, and so then we have here the electromyogram from the surface EMG, and then we measure the brain signals at the same time. So there's a nice coherence at about 20 Hz. For right hand contraction, it's over the left motor cortex. For left hand, it's over the right one. For foot, it's at the foot region. Okay, and then we know that this, we know about this coherence also that it's there only if the person keeps a steady contraction. If there's any ramp movement, so it's gone. And then this has led to the idea that this is Cortex-muscle coherence is some kind of index of stabilization or status quo of the primary motor cortex. And we also know that it reflects the outflow from the cortex to the muscles. It somehow, this first something happens here and 20 milliseconds later in the hand. So we put the subject to the shielded room, and he's keeping all the time steady contraction against this type of force transducer. And then he sees in the magnetically shielded room, so this is his view, he sees the experimenter in the same room with a similar type of manipulandum. And then this person is from time to time making PHASIC actions, like this. And this person is just keeping the steady contraction and viewing the other. And now we ask that, okay, we already learned that primary motor cortex should be activated while he's seeing these actions, but is something else happening? So, so this is a displacement of the experimenter's hand actions, and then we take also electromyogram from the subject that we see that he's not changing his contraction force. Then we look what happens in MEG, the MEG power in the primary motor cortex. So this is the frequency as a function of time, and this is with respect to the experimenter's movements. There's a clear suppression of this power is decreased, telling that, as we expected, primary motor cortex seems to be activated. But then when we look then this cortex-muscle coherence, that I said that it should be considered some kind of sign of the stabilization of the cortex. So we see that, okay, there's an, after this action, so in fact there's an increase in this cortex-muscle coherence, telling that it's somehow primary motor cortex is activated, but it's also stabilized more, and it's somehow strange. But we see that these don't occur exactly at the same frequencies. So our conclusion from this a little bit complex experiment is that motor cortex is both activated and stabilized during observed actions, but in different frequency bands, telling that most likely there are different neuronal populations.
How can you infer that this is stabilization and not response?
Yeah, because of the earlier studies that, for example, so we know that it's there only this. So this is like a person is keeping a steady contraction and then moving. So this is gone. So there's a lot of information that whatever somehow, if there's a jitter or whatever, so in the action, so this coherence is gone.
So you could see that in your last study in a trial-by-trial correlation, negative trial-by-trial correlation with actual movement, maybe.
Sorry.
So this could be correlated negatively with the measured person's movement.
Yeah, the measured person did not move.
No,
okay. Yeah, so that was the EMG control, electromyogram control from the muscles. Measured person did not move, but his motor cortex was activated, and then the other populations seems to show then this. sign of stabilization, so but in different neuronal populations. And interestingly, there's some monkey data recently from Roger Lemon's lab in London showing that primary motor cortex contains that kind of mirror neurons that are inhibited by actions. Okay, so just then to end, just one slide of my or 2 slides of my current work, which has been somehow going beyond these single person studies. So if we're interested about brain basis of social interaction, so we may say that, okay, we have presented more and more complex stimuli, even movies and different faces and whatever to people, but it's still something like unidirectional. Like we would be considering our subject to be just spectators of the world, not interacting or engaged persons. And that's why we've been, over the years, we've been building this type of MEG to MEG system. And of course, here's been important that this, it was a happy thing that this Elekta NeuroMag was a spin-off our lab, because then there are several identical devices many places. So we first connected one device with an identical device 5 kilometers apart in hospital, Meilahti Hospital. And this was just an audio link, so people were like able to speak by like telephone together, and then we locally saved the data, and we were also able to save the, of course, all the speaking information about the speaking. And this was just like an auditory channel, auditory connection, and we started to study already like a brain basis of conversation. And more recently, we have also added then a visual connection there. So we have audio-visual link, which has then a little bit longer latency. And for example, last summer we had about 1 week when we were collecting day and night data, so that the other person was in Erasme in Xavier's place and the other was in our place. And then something the people can do whatever there in these 2 places. And in the first cases we have been studying, for example, synchrony of motor actions. So we have also accelerometers from this guy and accelerometers from the other guy. Then we can have different instructions. They can be, one can be leader, other can be follower, or they can do that as they would like to do. And we can look at the synchrony of this, and finally we can then go to the look at the brain changes, but it's work still in preparation. So just to conclude that for the future, so at present we have devices, they are expensive and they can't be moved to different places, as we heard from Xavier, but they give us pretty good signals. So in the future, the wishes would be that we should have some good sensitivity from DC to 2 kilohertz, for example. Like in this case, these early studies, we had a stimulus that lasted almost 1 second, and we have a very nice, clear, sustained response here after that. And then I can just repeat what others were already saying. That's what I was as well thinking, that sensors, when they come close to the scalp, so they will increase, of course, the possibility to see this interesting spatial pattern, and we should have some adjustable array like in EG cap, so this is the wish. And then this whole scalp coverage is really important for artifact rejection, but also for reliable signals, so that you can go beyond this early sensory phenomena, but look at the sources of spontaneous brain rhythms and also signals that are related to different cognitive functions. So thank you very much.